Good day, my WholeMood students. So this is our fourth teachable, lectural, and tutorial video. In today's video, we are going to be discussing the use of energy, how cells use energy that they get. And before we get into this video, I would like to state that please hit that subscribe button, hit that like, hit that share, and hit that comment and tell me what you think about the video. I am a first year student at the University of the Western Cape, so it is what it is. Let's go. You ready? I know I am. A whole entire mood. So the vlogation. The use of energy. All cells capture and use energy. So now, what is energy? Energy is the ability to do work, and we find energy in many different forms, but we are going to focus on two types and forms of energy. We're going to focus on kinetic energy and potential energy. Kinetic energy, so let us say we have energy, which is the ability to do work. And then energy is broken down into kinetic energy and potential energy. Kinetic energy has to do with motion. It is the energy of motion. And then potential energy is the energy that has to do with the position of an object or atom or molecule or whatever you're dealing with at that particular moment. And then the total amount of energy is the sum of the kinetic energy and potential energy. We are going to move into thermodynamics. Thermodynamics, it is the study of energy transformation. It is the study of energy transformation. And then we are going to look at two laws of thermodynamics. The first law of thermodynamics and the second law of thermodynamics. So thermodynamics has two laws, right? There's the first one and the second one. So now the first law of thermodynamics states that energy in the universe is constant. Energy cannot be created or destroyed, but it can be transformed into one form to another. So what that means that energy can be transformed into kinetic energy to potential energy, and energy cannot be created or destroyed, and energy in the whole entire universe is constant and stays the same. That is the first law of thermodynamics. And the second law of thermodynamics states that the amount of some of the energy is lost through the environment as heat, and energy lost as heat can never be gained back. And this is due to the random molecular movement of atoms and molecules. And then now energy lost as heat is disordered, and then now we get the term called entropy. And entropy is the measure of this randomness, which is the random molecular movement, and is also the measure of this disorderedness. So now energy lost as heat increases in entropy, and entropy is always increasing, it is never decreasing. So that is the second law of thermodynamics. The first one tells us that energy in the universe is constant, and then the second one tells us some of the energy is lost to the environment as heat, and energy lost as heat can never be gained back. Then we have chemical reactions that sustain life. Within your body, there are multiple chemical reactions that are happening at the same time. So we have chemical reactions. Within your body, there are multiple chemical reactions happening at the same time that keep you alive. Without the chemical reactions, your body would not be able to do the work. So then all these chemical reactions, we group them together and call them metabolism. So metabolism is all the chemical reactions in your body that keep you alive and make sure that your body performs its functions. And then now, remember that a chemical reaction is when reactants come together and react, and they come together and they bond to create new, completely different molecules, which are called products, right? Chemical reaction is bringing reactants together to form products. Metabolism is all the chemical reactions in your body that work together to sustain life, right? And then we have different types of reactions. We have endergonic reactions and exergonic reactions. So now I am going to write a very simple sketch of each. So before I write the sketch, let me explain what they are. Endergonic reactions are reactions that require an input of energy. They require an input of energy for the reaction to proceed, and then the energy of the reactants is always less than the energy of the product. The energy of the reactants is always less than the energy of the product, and then they build bigger molecules, complex molecules from smaller molecules. Excuse me. And then exergonic reactions are essentially the opposites of endergonic reactions, whereby a very short supply of energy is needed for the reaction to start, and then the products have less energy than the reactants. And in this one, it is used down to break bigger complex molecules to smaller products, right? To smaller, much less complex molecules. And then now a small representation of the of them. We're just gonna have this. You're gonna have this. And then if you look at the two graphs, you can see that the exergonic one, the endergonic one, we have reactants here. Then we have products here. You see that your reactants have less energy than the products. So now for the reaction to start, we need to give it a boost, a push, whereby, as you can see, there's this very big lump. This very big lump is called activation energy. This lump over here is called activation energy. Activation energy is the amount of energy needed for a reaction to start. It is the amount of energy needed for a reaction to start. Without activation energy, the reaction will not be able to start. Then after the reaction has started and activation energy has been undergone, you see that we get products which have more energy than the reactants, right? This is the reactants, this is the product, okay? And then now we come to the exergonic one. The exergonic one, you see that your reactants have less energy. And as you can see that even the exergonic reaction, it needs an activation energy, which is a big lump, for the reaction to start. But it does not need that much energy, but it does need that initial kick, that initial boost that initiates the reaction. And then after that, you see that the energy of the product is less than the energy of the reactants, right? So now for the endergonic reaction, energy is absorbed because the products have more energy. And then now for the exergonic one, energy is released because the products have less energy than the reactants. And then the energy that is needed, the energy that is needed to kickstart this reaction, it comes from a molecule called ATP. A molecule called ATP, which we are going to get into very now. ATP is very important. And then now we have redox reactions. Redox reactions are reactions that oxidize one molecule or atom, and then they also reduce one molecule and atom at the same time. So then now we have reduction plus oxidation, which obviously comes with redox reaction. So redox reactions are also called oxidation reduction reaction or just simply redox reactions. So now oxidation means that the loss of electrons from one atom, a molecule or ion. Loss of electrons, and then reduction means gain of electrons from one ion, molecule or atom. So oxidation is loss of electrons and loss of energy, loss of electrons and loss of energy. And then reduction is the gain of electrons and the gain of energy at the same time. So now for an oxidation for oxidation to happen, a reduction must also happen. So oxidation and reaction happen at the same time, because once a molecule is oxidized, the other one is reduced at the same time. So they always happen at the same time. They never happen at different times. Okay. And with that said, is anything, am I missing anything? Yeah, yes. And then now oxidation reduction reaction is also called redox reaction, give us an electron transport chain. So now the electron transport chain is a group of proteins, a group of membrane proteins which are found on a membrane that shuttle electrons from protein to protein. They are a group of membrane proteins that shuttle electrons from protein to protein. And remember that reduction and oxidation has to do with the gain and the loss of electrons. So now the electron transport chain transports those electrons. And so for in the electron transport chain, so the electron transport chain is like this. This is your membrane, and then this are your proteins. This is protein number 1, protein number 2, and that's protein number 3. So now an electron will come here, do its job, then leave here, then come here, do its job, and then leave here, and then come here, then do its job, and then leave. So now these electrons are moved from this membrane, from this protein to that protein to this protein. They are transported within the proteins. And obviously now a protein is first reduced because it first gains an electron. Then once it gains the electron, it loses the electron. Once it gains the electron, it loses the electron and it becomes oxidized. And then with each movement, with each transport trait, with each protein, for the electron to move to the next protein, energy is released, and then the energy that is released, it moves down, and it makes a gradient, right, which we'll get into the next topic when we talk about respiration. But just know that when these electrons moves, they release energy, which creates a form of potential energy, and that potential energy is a form of a gradient. Okay. And then now with that said, we are going to talk about ATP. Like I said, ATP is it is a type of RNA, in a sense that it is composed of a 5' sugar, adenine, nitrogenous base adenine, and it has 3 phosphate groups. And then ATP stands for adenosine triphosphate. So adenosine comes from the adenine that it gets, and then the triphosphate, it comes from the fact that it has 3 phosphate groups. And then the ribose, it tells us that it is a type of RNA, a very special kind and type of RNA. ATP, most of the ATP is produced and it originates in the mitochondria of cells, and it is very, very responsible for providing energy that cells need to perform their job. Without ATP, nothing can happen. Life cannot function. So ATP is very, very important, and we need to know that. And then we have enzymes. So now enzymes are also very, very important. They're also very, very significant. Enzymes are organic molecules that catalyzes chemical reactions, which means that they speed up the rates of a chemical reaction and they lower the activation energy. The activation energy, remember that it is this, okay? So now enzymes bring reactants together faster so that the reaction can occur at a very, very occupied rate. So now remember we had this now. So now you see the activation energy is high. So what an enzyme does, it comes in and it lowers the activation energy, and then the reaction occurs at a faster rate. You can see that this one is lower, meaning that the activation energy is lower and the reactants come together faster and the reaction proceeds faster. It's like climbing a hill in a mountain. It's going to be easier and faster to climb a hill, but it is going to be harder and slower to climb a mountain. So like this is the hill and that's the mountain. And then the activation energy, the enzymes lower the activation energy so that reactions come together faster, okay? And then enzymes are substrate specific. So let's say this is an enzyme, and by the way, enzymes don't get used up in a chemical reaction. They come together, they perform their job, and then they bring reactants together, and then once the reactants are done, they release the reactants as products, which are new products that are formed, but then the enzyme itself stays exactly like that. So then now the enzyme has a part called the active site. The active site is where the substrate or the reactants bind to it for the reaction to happen. So now this thing here over here is called the active site, right? And then now your substrate, which is also called the reactant, is gonna come together with the exact same shape as the active site. Only substrates with this exact same shape can bind to this substrate, right? To this active site. If it doesn't have this shape, it doesn't bind to the active site. And then once it binds, the reaction happens, and then when the reaction is done, the substrate, let me put it down here, the substrate, which is also called the reactant, it leaves the active sites. The active sites unlocks it, and then it leaves as new products, which are which are new formed molecules or compounds, right? And then now let's say that we have a lot of concentration of the active sites and not a lot of concentration of the substrate. So what's going to happen is the substrate is going to bind to the active sites, but then the reaction is not going to be occurring at its fastest rates and its best rates, because now you have more concentration of the active sites and less concentration of the substrates. But then if you increase the concentration of the substrates, the reaction occurs at a faster rate, you get more products, and the reaction reaches its optimum. Then once the reaction reaches its optimum, adding much more substrate concentration is not going to increase the reaction, because now we have reached maximum. We're not going to yield much more products to get it. Yes, that is what enzymes do to us. And then we also have non-protein co-helpers that work with the enzyme. Those non-protein co-helpers are called cofactors, and the cofactors are substances that are needed for the enzyme to perform its job. And just like the enzyme, they catalyze the reaction and they do not get used up in a reaction. Without a cofactor, the enzyme, they cannot do its job properly. Okay, so yeah. And then now we have membranes. So now a membrane usually it is called a biological membrane when it is a phospholipid bilayer, which we already discussed. A phospholipid bilayer allows nonpolar substances and lipids to pass through the bilayer without the use of a protein, and then polar substances need the help of a protein to move through the phospholipid bilayer. So now the phospholipid, like I said, it is selectively permeable. You also get something called a concentration gradient. The concentration gradient is when the solute concentration is bigger on one side than in the neighboring side. And then now, obviously, concentration is always the concentration gradient will always dissipate unless energy is expended to sustain it like that. So what that means is that concentration gradient, the solute will move from where it is more concentration to where it is less concentrated at any given point. Alright, so now we are going to talk about mechanisms and transport systems that allows for the movement of the solute concentration from where it is more concentrated to where it is less concentrated. So we have passive and active, right? So both of these are form of transport systems. So now we're going to start with the passive transport. The passive transport, it is the movement of a solute, of a solute concentration, the movement of a solute from where it is more concentrated to where it is less concentrated. The solute moves from where it is more concentrated to where it is less concentrated. And then we have diffusion. All passive transports have to do with diffusion, and in diffusion it is the movement of solutes down its concentration gradient, which means it is moving from where it is more concentrated to where it is less concentrated. So then now diffusion has simple diffusion, let's put it down, simple diffusion, osmosis, and facilitated diffusion. So now simple diffusion, it does not require proteins, whereby it does not require energy, and it just the solute moves from where it is more concentrated to where it is less concentrated, and there is no energy required and there is no proteins required. And then we get osmosis. Osmosis is the special movement of water through a selectively permeable membrane. So now it is when water moves from where it is more concentrated to where it is less concentrated. And then facilitated diffusion, it is when we require proteins to help the solute moves from where it is more concentrated to where it is less concentrated. As you can see, all this passive transport, all these diffusions, it is just the movement of solids from where it is more concentrated to where it is less concentrated. In simple diffusion, there is no proteins required. In osmosis, it is water, and in facilitated diffusion, it is when we require proteins to help the solute moves from where it is more concentrated to where it is less concentrated. As you can see, all this passive transport, all these diffusions, it is just the movement of solids from where it is more concentrated to where it is less concentrated. In simple diffusion, there is no proteins required. In osmosis, it is water, and in facilitated diffusion, it is when we we use proteins, right? So then now in simple diffusion, usually the substances that moves are the non-polar substances, and then in facilitated diffusion, the substances that moves are the polar substances that cannot move to the selectively permeable membrane, so they require the use of transport proteins, which help the system move down its concentration gradient through those proteins and then move into the other side where it is less concentrated. And then from there we have active transport, is the movement of solids from where it is less concentrated to where it is more concentrated. So obviously for it to move from where it is less concentrated to where it is more concentrated, we need energy to do that. It is not moving down its concentration gradient, so we need energy, and usually we get this energy from ATP. ATP is a very hardworking cell. It is, well, not cell, but then it is a very hardworking molecule. So it needs energy from the ATP, and then it also requires help from proteins, and then it moves the solutes from where it is less concentrated to where it is more concentrated. And then now the last part of this topic, we are going to talk about endocytosis and exocytosis. Okay, so we are going to talk about, let's use a blue pen. We are going to talk about endocytosis and exocytosis. So now endocytosis, it is when molecules, solids, and substances move into the cell, right? That is move into the cells, like inside, through the membrane. And then exocytosis is when substances, molecules, solids move out of the cell to the cell membrane. Like exo means like X as in exit, right? X as in exit. That is how I remember it. So then now what happened in endocytosis, a vesicle within the cell, a vesicle is created, and the vesicle encloses all the substances that need to move out of the cell, and then the vesicle attaches to the cell membrane. So now we have something like this, where we have a vesicle with all the stuff that need to move out of the cell, and obviously this is now the cell. All the substances that need to move out of the cell, they move to the cell membrane and attach to the cell membrane. Then after attaching to the cell membrane, the vesicle stays with the cell membrane, and then the substances are exported into the cell. Wait. I'm really sorry. I just explained exocytosis, whereby the vesicle is made inside the cell, and then the vesicle touches the cell membrane, and then it transposes, takes out the other substances out of the cell. And then endocytosis is the opposite, whereby the substances move into the cell membrane, and then they attach to the cell membrane, and then they form a vesicle, and the vesicle moves into the cell, and the vesicle becomes part of the cell. That is endocytosis. So now endocytosis, substances go into the cell, right? Exocytosis move out of the cell, right? Yeah. And this is the whole mechanism. And then we have pinocytosis. Let's say pinocytosis and phagocytosis. Pinocytosis, it is the movement of small molecules, small substances, and small solids into the cell. And then phagocytosis, it is the movement of bigger molecules, bigger substances, and bigger solids into the cell. So both of these are part of endocytosis. And remember that endocytosis, it is when substances move into the cell, and in exocytosis, it is when they move out of the cell, right? And remember that a vesicle helps carry those substances into or out of the cell, and they move through the cell membrane. And that is our topic of the cells use energy. Thank you very much for watching this video. I really hope you gathered some information that will help you do your test, that will help you do your exams. And please practice, work hard and push hard and grind hard, and you will get those results that you need. I really appreciate you watching the video. Thank you, and I'll see you in the next video. The next video is going to be about photosynthesis.